Okay, we should probably make a start. Welcome everyone to the 5th seminar organized by the ESIP-funded Network for the Integration of Speciation Research. The aim of this network is to integrate speciation research in 2 ways. So firstly, we want to bring speciation researchers together through this seminar. And then secondly, we are building a database for integrating and sharing data on speciation. Okay, so if you are new to this, check out the IOS website for details and also instructions on how to sign up for email alerts. This seminar will be made available as a recording in our YouTube channel. And today's seminar is part of the 2nd series of seminars on understanding and measuring barriers to gene flow. And the topic today is the role of geography in generating reproductive isolation. We have 2 great speakers, Clarisse Palma-Silva, I hope I've pronounced your name correctly, and Rosemary Gillespie. Before I introduce our first speaker, just a quick reminder how this seminar is organized. So each speaker has 20 minutes to talk. There's time for a few questions after each talk. And then we have a bigger discussion an hour from now of 30 minutes. So we'll save some of the really good questions for that. If you have questions during the seminar, please write them into the chat. Okay, so without further ado, it's our great pleasure to welcome our first speaker, Clarisse Palma-Silva. Clarisse is a professor in the Department of Plant Biology at the University of Campinas in Brazil. She obtained her PhD in genetics from the University of Rio Grande do Sul. She did a postdoc, several postdocs, but all in very close succession in Kew Botanic Gardens and the University of Brazil, had a fellowship in Switzerland, and is now full professor at the University of Campinas. Clarisse studies evolutionary biology in neotropical plants, especially bromeliads and orchids. And she's particularly interested in local adaptation and the evolution of RI in these systems. And she's going to tell us about evolution in naturally fragmented habitats. So please, Clarisse, whenever you're ready, we're really looking forward to your talk.
Good, sorry. Just because I choose the pen, then I could not start the sound. So first of all, thank you very much for the nice introduction, Conrad, and for the invitation. I'm super happy to be here. I love the topic of speciation and geography as well. So I'm really, really delighted to be here to be able to not answer the questions, but making more questions and trying to collaborate with our thinking and understanding of how reproductive isolation evolved. So I'd like to thank, of course, the organizing committee of the Network for Integration of Speciation Research, the ESEBI. This is a very nice initiative, and I hope it will last for many, many years. Super exciting. So let's start with this, with my talk. Okay, so I cannot go a little bit there. Maybe here, not very nice, but okay. So yeah, the title of my talk will be a little bit general, the evolution in naturally fragmented environments. As Conrad mentioned, I really enjoy this special topic. And I'm a professor at the Evolutionary Ecology and Plant Genomics Laboratory, together with Professor Fábio Pinheiro, is my husband. And not only the life, but then we share all the academic interests, including speciation. And today, I would be happy to think and talk. It was really nice to prepare this talk actually, because it made me thinking a lot about the whole of geography and speciation and species formation. I'm gonna do like that. And so the idea about geography and species formation is quite traditional, quite classical. This is why I put the Galapagos in here. Everybody studying that or even studying biology will be aware of the idea that geography will somehow promote speciation. This is because geographic isolation will produce interruption in the gene flow, and isolated population will diverge both morphologically and genetically. So then this is a very easy idea, very obvious, I would say, but then there's a lot of considerations that and complications that we have, and that we will discuss today, starting with, does speciation really require, all the process of speciation require geographic barriers? Okay, so geographic models of speciation have been also very widespread in the study of speciation. So we have allopatric, parapatric, sympatry, and then these modes of geographic speciation are actually a continuum of the idea of dispersal and habitat choice, niche evolution of the organisms. But there are some difficulties here, because these are dependent of a scale, a geographic spatial scale. So something we call sympatry or sympatric species or sympatry occurrence may be a micro allopatric. And this is a little bit complicated, not only because of that, but because the process of speciation, as I see, or most of us, is a continuous process. And when we try to characterize or to classify things that are continuous, we will have problems, we will lose information on that. So then another complication is that geography not really, it's the best indicator of the presence of the gene flow or lack of gene flow during the process of speciation. Here. And the other point that when we think about allopatric speciation, usually we think about a sudden process with a complete lack of gene flow, but we know that even this process can be in agreement with the continuum of speciation. Most of the geographic barriers, mountains, continent separation, will be actually a gradual process, millions of years. So during a certain time, now I can do that. During a certain time, we can have some gene flow that will decrease during the formation of the barrier, and then when the barrier is established, is complete, then we will have the complete differentiation or a complete stop of gene flow. And then, in fact, it will be promoting more divergence across these populations. So actually, with all these complications and concerns about how we study geography and how geography can help us to understand speciation, maybe this is why we are here today, right? And maybe it makes more sense to investigate the gene flow and natural selection and the evolution of reproductive isolation than trying to classify them. And this is what we would like to do. I think it's the best way of studying geography, understanding how geography will promote speciation and reproductive isolation. So trying to use some examples of what I am doing, what we are doing in our lab, I would like to introduce you the ideas of the naturally fragmented habitats. We usually think about islands that are the most classical example, but many other things that could be considered naturally fragmented habitats. They are actually quite common in nature, and this is why we are concerned on their role in the speciation. So it could be a mountain, could be lakes, as we have here, could be riparian forests that are different from the matrix surrounded, and the rivers, could be rivers itself, and could be many other things, including inselbergs that are the ones that we like very much to study. The inselberg are those rocks, those granitic or gneissic rocks that are surrounded by a matrix of forest, completely different from what we have in the rock. So it makes them resemble an island, a terrestrial island as well. So studying all these example in naturally fragmented habitats will maybe help us to understand a little bit on the whole of the geography. So naturally fragmented habitats has two important aspects in the evolution and the diversification. For one hand, we know that extinction and then reduced population size that usually happen in such habitats may lead to local extinction, will promote low efficiency in natural selection because it will increase also a drift, and maybe promote lower and decrease, extinction. And for the other hand, we know that speciation is also important in such habitats because lack of gene flow will promote population differentiation, and such process is super important for the speciation itself and will increase speciation. So here we have this question, what we should expect, high or lower levels of diversification in such habitats. So it's a balance, trying to understand each case and which situation we have. And then to talk a little bit more about these issues on geography and diversification, reproductive isolation, I separate my talk into points, into steps. The first one, I would like to talk especially about the interspecific gene flow, and then how reproductive isolation will be evolved in allopatry. So we know that geographically isolated population will differentiate. This is the process, but it's important also to understand that they need to persist until the evolution of reproductive isolation and the ecological divergence until the completeness of speciation process. So this is one aspect which is, in my point of view, very important for understanding the evolution and the speciation of such habitats. And the other one, which is not completely separated, but it's nice to separate here to exemplify better, is the interspecific gene flow, when it happens in sympatry and parapatry. And then maybe it could be a consequence of secondary contact or even speciation gene flow in situ speciation. So both of this process are very interesting and important to be discussed in the context of evolution and reproductive isolation in naturally fragmented habitats. So we start with the first part. We have this beautiful bromeliad species. Bromeliads are the same family of the pineapple. And then Pitcairnia flammea is a species complex with a huge morphological variability and super different. Unfortunately, I cannot show you all the differences we have here for sake of time, but then it's super different. They have several species and varieties and taxonomic complications. So all the schematics and taxonomies get crazy with this group, but then when you have such a craziness in taxonomy, you see the opportunity for studying the process of speciation occurring, undergoing in the time. So it's very exciting to study this group because they are a very good example of a speciation. So what we have here in this map, all this is a part of Brazil, right? The eastern Brazil, you can see here Brazil and South America. The gray shade is Atlantic forest, and the dots are the currents of the species where we collect them, or we have get them from GB for species link. So where people have found the species. So it's wide distributed, as you can see, to more than 2,000 kilometers. And then we collect from the 25 population, the red stars, okay? I forgot to say that this is, although this is a very morphologically and complex species group, it's a monophyletic plate. So it makes us very happy to be able to study this important clade in the group. The genus has more than 400 species, which is quite impressive. They diversify in less than 12,000 million years, which is very fast as well. So just briefly showing some results we have in this first screening on diversity and genetic structure. All each population, as you can see, has a different haplotype. So it's a super micro structure, genetic structure on this in this population level. You can see here the network. It doesn't have very good definition in haplogroups or something like that. And then this fixation of haplotypes are super interesting. And the same thing we found for the nuclear markers. So using microsatellites, in the time we use that. So from out of this 25 populations, we found 22 genetic groups. Basically, again, each population, each rock is a different genetic group, has a different gene pool, with some exceptions here and here, very few exceptions. So this complication, right? Of course, there was a question, what is a species and how can we delimit that? So we use this coalescent analysis for species delimitation, and out of the 25 populations, we found 24 operational taxonomic units, suggesting that most of the populations are evolving independently. The group evolved 2 million years ago. They diversified 2 million years ago, suggesting that the Pleistocene climatic changes were important for such diversification. Oops, it's blocked. I don't know why. Okay. So then other important considerations in these results is that although there's extremely high population differentiation, we found absence of isolation by distance. And actually this is what we expect for this highly isolated habitat. Doesn't matter if the population is close or far, they are super genetically differentiated. So this is why we do not expect isolation by distance, which is kind of a counterintuitive, I know. And one important process here that may be underestimated is the long distance dispersal. So here is the picture of the seed. It's a super small seed, autochoric seed dispersal syndrome, doesn't have any appendage. All the 400 species for the genus doesn't have any appendage for dispersal, suggesting that this long distance dispersal will be important. But this is also important to highlight that long distance dispersal is a random process and is also sporadically, very rare process. So it does not account for connectivity, right? So long distance dispersal doesn't help to maintain gene flow among populations. It helps during the colonization process. So this is when this process, this long distance dispersal will be important during the colonization. So this is maybe why we are able to see such a geographic range, large geographic range for the group. And one other point that help us to think that is that the species are self-compatible and very tolerable for high levels of inbreeding. And in fact, inbreeding coefficients are super high, up to 0.4. So, okay, so we have this first screening of completely geographic differentiation, extremely genetically variable and morphological differentiate populations. And then, but how can we understand, in fact, how the species are evolving, how the populations are evolving and becoming a different species, right? So this is one important point, I think, in the process, is to understand geography and environmental heterogeneity at the same part of the process. So to understand local adaptation, right? We will be able to understand local adaptation in a group that have strong drift and limited gene flow. And this will be very important for promoting reproductive isolation and speciation. I forgot to start the, what I need to do. So those are the populations we decide to start in the second part of this study. So here we have this small part here of the Atlantic Forest, the Brazil. It's close to the city of Rio de Janeiro. Here is the city of Rio de Janeiro, and here is Niterói. And then we collect different populations, so a small number compared to the other study. And in an elevational gradient of 2,000 meters, some populations are up to 2,000 meters, which is the highest for this mountain system, from these mountains. And then some population at sea level, and for other populations that you can see in the bottom of this map, in a gradient of this elevation. Okay, so what we found first here is that we tested, Kleber Chaves tested as a postdoc in our lab. He tested the thermal tolerance for heat and cold stress, and we could see actually that we found significant differences in the tolerance and the thermal tolerance for populations in up mountains, 2,000 meters, and the population in the sea level. And actually, this is what we are understanding now in this system, is that isolation, geographic isolation, will, despite strong drift, facilitate local adaptation, because now, because we don't have the gene flow, or less gene flow with different and not adapted alleles among these populations, it is also easier to promote this local adaptation, and that will also, I guess, promote reproductive isolation. We also analyze some functional traits that may be involved in the local adaptation of these populations, and we see some traits like succulence and leaf area and trichomes are larger in the populations that occur in the sea level, and some other traits are larger and are more important for populations in the top of the mountain, like the abaxial trichomes that will promote more resistance to UV radiation. So altogether, these indicate us that not only geography are interesting, but then local adaptation in this heterogenic environment is also important for the speciation process. So talking about now the second part about the interspecific gene flow when the populations are in the same places. We study 3 of these populations, 3 of these 25 populations were sympatric populations with more than 1 species, and 3 or 2 and varieties in the same population. Just briefly showing you that for some of these sympatric populations, we could not see gene flow. We could not see even hybrids. Here we can see hybrids, but just F1s, which indicate, not showing here, but indicate that we do not have introgression. And then one population, we found hybridization and introgression. So it indicates that we might have secondary contact, because very likely those populations are never in contact. It was a secondary contact, but somehow they could speciate in situ, right, with gene flow. So here is another example with the same species from the same genera, and I studied it during my postdoc. We could see a lot of hybridization, introgression in the speech cornea that happens in the same rock outcrops at the city of Rio de Janeiro, and extremely low interspecific, again, very low interspecific gene flow among this species and the population of this species. And then we also could see after doing a ABC analysis that speciation with gene flow, more gene flow in the past and less gene flow in the present, will indicate that this species is probably under this idea of speciation with gene flow in situ speciation. So I hope that the idea of understanding intraspecific gene flow and intraspecific gene flow during speciation is not actually an answer, but then another question, right? What I show here is that we have some variable results and patterns of speciation and reproductive isolation. They are different among different populations, which is an agreement with this continuum of speciation, this species complex, and also open the question and the idea that maybe they have a positive evolutionary impact because introgression in such a limited gene flow populations will maybe interesting for the evolution of the species. So I hope I didn't not took all my time, and I'd like to thank all this the financial agencies that finance the grant, our study, and the students and researchers that were involved in this project. Involved are those that work directly in this data. And with that, I would like to thank you and to ask you if you have some questions or answers to our study. And thank you very much.
So we have a little bit of time for questions, but maybe one or two. So, I mean, one question that I had just at the end when you showed these modeling results. So you seem to be suggesting that a model with gradually decreasing gene flow fits best. Is that, to what extent is that in contrast with the idea that, you know, these are just mainly in allopatry, as you sort of explained at the beginning? So is there different signals that you're picking up through the demographic modeling?
Yes, because we tested here the completely allopatry, right? And then we test this attracted decreasing in gene flow. This was the idea. Actually, it was a little bit fast. I know that's a short time, but then the point actually is also the fact that some species are not sister species in the phylogeny. And they do not, and these are sister species. Maybe it will be interesting and better to say that when we have this speciation with gene flow in the sister species and the other population, they are not sister species. That maybe help to understand in the context.
Yeah. So we have one question here in the chat. Where you have genetically distinct groups within patches, are they adapted to different niches? So that's from Roger Butlin. And maybe related to that, what do they do during ice ages? Do they go up and down mountains, or how persistent are these habitats, these fragmented habitats?
We don't know that. Yes, this is something we would like to know very much. We are working for that. And we don't know what happened, if they were fixed, or if they expand, or if they move their range, their niche range. We don't know that, Roger, yet. But I hope next time we will be knowing that. We are working on the whole genome sequencing for trying to understand a little bit of demography, because with the sequences, Sanger sequence, we could not understand much of that. Any other questions?
So if there are other questions, please post them in the chat. We have time later on during the discussion. So we have another half an hour after the second talk to chat. So don't forget to post questions in the chat. And it's probably time to introduce our second speaker now.
Great, thanks. Yes, thanks, Clarisse, for this great talk. And now it's my great pleasure to welcome Rosemary Gillespie, who is our second speaker today. Rosie grew up in Scotland and graduated from the University of Edinburgh. And then she moved to the US for a PhD at the University of Tennessee, where she got really excited about spiders. After 2 postdocs and a position as assistant professor at the University of Hawaii, she became a professor at the University of Berkeley, where she is until now. She's also the director of the Essig Museum of Entomology and previously acted as the president of the American Genetics Association and the International Biogeography Society, just to name a few. Rosie is well known for her work on the evolution of species communities on hotspot archipelagos, and her main focus is on spiders on the Hawaiian archipelago. These islands are super interesting, as I'm sure we'll hear in the talk as well, as they range in age and thus provide great opportunities to compare older and younger species communities. Rosie has used these to shed light on how species communities assemble and how island radiations unfold over time. And today, Rosie will show us some of the insights she gained there about the role of geography in speciation. So Rosie, when you're ready, please just go ahead and share your screen. We look forward to your talk a lot. Please go ahead.
Well, thank you so much, Johanna. Let's see if I can get this to work again. We can see if, does that show good or bad?
Fortunately, the wrong way around. Perhaps you can, on top left, there's like a little circle. Maybe you can just click on that. Yeah, perfect.
Yep.
Okay, just a sec here. Oops, sorry, I just need to get, yeah, for whatever reason, it hides. Well, we're all set here. So first, yes, thank you so much for having me here. That's a true pleasure. Thank you so much for introducing me, Johanna. And thanks, yeah, this is a great forum and I'm really looking forward to the discussion already. But what I want to do today, and Clarisse has set this up just beautifully, but what I want to do is just talk about the speciation focusing on islands. And so I'm going to go through fairly quickly the big picture of speciation on islands, because Clarisse has already given you a really nice broad overview. And so just to start off with, the big thing about islands is their isolation. And islands, I mean, so lakes can behave as islands. And as you heard from Clarisse, you've got these fragmented environments that can behave just as islands as well. And so this kind of attribute of islands has been used for a very long time. The isolation of islands was really what gave Darwin many of his insights in figuring out how natural selection operates. Darwin, of course, was working in the Galapagos Islands, and at the same time, Wallace was thinking about these issues based on the islands of Indonesia. And so we think, well, you know, what makes islands special? I should also mention that it was islands that formed the basis of other major influential theory in our understanding. But I won't talk about this so much, the theory of island biogeography, but it's critical for our understanding of species area relationships and things like that. But what I want to do today is focus on speciation, and I'll just very quickly go through the ideas of allopatric, parapatric, and sympatric speciation on islands. And first of all, just thinking about these 3 scenarios, just quite quickly, what we know about, you know, isolation and how organisms speciate on islands. One of the, you know, the nicest examples of speciations and diversification is where you see the breakup of Gondwana. And so Gondwana, the southern continent, broke up and pulled the different fragments with it. And as this is an example, as Johanna said, I work on spiders, and so you'll see many examples from spiders, but this is one of my students was working on these pelican spiders. And so she showed that these spiders had diversified with the breakup of Gondwana, and so formed different species or different species groups as these continents broke apart. But what I'm going to do today, is focus more on oceanic islands and islands that are hotspots, and look at how you can get allopatric speciation in these different hotspot islands right across the world. You already heard from Clarisse about the Galapagos Islands, and I'll be focusing mostly in the islands of the Pacific, but very mostly on the islands of Hawaii. So many of these islands are hotspots, and so they're arranged in a chronological order, and so I'll be highlighting that. And just to show you one of the very nice examples of how islands provide isolation, this is an example again from one of a student in our group, and Ellen Claridge, and Ellen was interested in how speciation had happened across the islands of the Pacific in this group of Rhynchogonus weevils. And what she was able to show was that these weevils have basically island hopped and just diverged in isolation as you go from one island to the next. So basically they'd hopped, just going from one archipelago to the next, and then hopping down each island chain from the older to the younger islands. So these are just really nice examples of allopatric speciation. But as I said, I want to focus on the islands of the Hawaiian chain. And first of all, this is a hotspot archipelago, so it's arranged from older to younger islands. And because of this chronology, you can get at some really interesting things. And just to highlight some of these aspects, this is Carrie Shaw's work, and we did a review just not that long ago, just to figure out this idea about how organisms just jump from the older island to the younger island in the archipelago, and they speciate as they go from older to younger islands. Why do they do this? Because the trade winds run perpendicular to the islands, and there's no apparent reason except for the age. And what seems to be happening is that you probably get founder effects with taxa go from one island to the next one as it forms, and then they monopolize it. It's basically you're basically getting monopolization of those initial founders, and they making it much more difficult for subsequent colonists to get in. So this tendency for taxa to go from older to younger islands is very common in hotspot islands, and it gives you a nice kind of insight into older and younger speciation events. And this kind of effect was used in really detailed, beautiful studies of allopatric speciation, in particular in Hawaiian Drosophila. This is the work of Hamp Carson on the left and Ken Kaneshiro on the right. And they showed how Hawaiian Drosophila have basically hopped down the island chain. And as they've done so, they've diverged in isolation by and large. And it seems that in each little population, you get very different reproductive behaviors, and it seems to be divergence in reproductive behaviors that is associated with speciation in this group. Okay, so that's looking at allopatric speciation. These islands are also really quite nice for looking at parapatric speciation. And I just want to give you just a preliminary look here at this wonderful story of these wolf spiders here. This is Lycosa hawaiiensis. This is a wolf spider, and you see here it's a wolf spiders. This is a mom wolf spider with all her babies on her back, and so you can see she's got buckets of them just all attached to her there. And the other thing about wolf spiders is they've got very large eyes, and so you can see here they just have these very large eyes. They're nocturnal. They accentuate the light, and so they can, you know, see well at night. So why am I talking about this in the context of parapatric speciation? Well, these animals have gone down caves. And so this is on the Big Island of Hawaii. And so they've gone down caves on the Big Island. So here you're looking at Hawaii, the youngest island. And what's happened is they've lost their eyes. If you look at, or at least they've become really reduced. If you look closely here, you see very small eyes. So there's really very little, they're just very reduced. They've got lighter in color. And so very clear example of the environment pulling these organisms in different directions, such that the cave species has its closest relative on the surface, and the ones in the cave have clearly diverged. So then the other cool thing about this is you come over to Kauai. And Kauai, they've done just the same thing. And you say, well, why is that so cool? You know, I've just told you about parapatric speciation down caves. Well, they've done the same thing, but they've had 5 million years to do it, as opposed to being very young on the youngest island. So with that long extended period, then you look at this wolf spider here. Here's a mom with just, you know, they have very few babies when they're the cave species. And if you look at this mom here, look at does she wear her eyes? She has no eyes at all. This is commonly called the no-eyed big-eyed spider. It's completely lost its eyes, just because there's a much longer time for speciation to have happened. Okay, so that I've talked about allopatric speciation, parapatric speciation. I'm just going to give you a couple more insights here, just and very summarily. Islands have also been the place for looking at incidences of sympatric speciation. And in particular, this is the work of Savalon et al. And they looked at divergence, sympatric divergence in palms on Lord Howe Island, and were able to show that these palms had diverged into different soil types. And they did a genome scan, showed that few genetic loci are more divergent between the two species than expected under neutrality, consistent with the idea of sympatric speciation involving disruptive or divergent selection. Another example was one that was suggested by Marta Barluanga et al. And they were looking at Midas cichlid fish in Nicaragua, and they were able to show that these cichlids diverged into caves, into different parts of the lake, and so basically they were pulled apart. They diverged along the limnetic benthic axis. And so you say, well, that's quite cool example of sympatric speciation. I would argue a little bit that maybe it's almost like parapatric speciation, but we can leave that there and just think a bit more. What I want to do now, rather than just dissect out sympatric versus parapatric and allopatric, I want to just focus a little bit more on Hawaii and using the Hawaiian Islands as a chronosequence. So what I want to do is just look specifically at the chronology here and give you just a couple of examples of how we can use the chronology to look at the speciation process. So what I want to do first of all is look at these hotspot islands, the very youngest of the hotspot, and focusing on Hawaii to understand what is the first initial stage of diversification. And so we're going to start on very, very young islands. And this kind of comes back to what Clarisse was talking about, naturally fragmented habitats. So I'm first going to look at this from a community perspective. So thinking about this, the Big Island of Hawaii is a naturally fragmented landscape. And so you think, well, you know, all of basically it's islands within islands. So what's happened here is that the lava has come down, fragmented the forest. And so you get little patches of forest of different age. And these are called kipuka. And so this perpetual fragmentation, isolation has allowed speciation to happen. And so this is a study that one of my students did a while ago. She was looking to what extent do these lava flows obviously they're creating patches and isolated patches of habitat. To what extent do they prevent or limit gene flow of populations in the forest fragments? And so she focused on this. I should say that this idea is not is something that Hampton Carson himself came up with a while ago. The idea that this young island, or the dynamics of the youngest stage of the archipelago, served as a crucible of evolution. So basically, it's forming this kind of fomenting pot of isolation, separation, before things come back together again. So Amy, a while back, looked at this, the effect of this isolation. And what she was able to show was that looking at species that you find within the main part of the forest, within the forest, what she found is if you look at divergence, so the x-axis is distance and the y-axis is looking at FSTs for mitochondrial DNA and for allozymes. Those are genetic markers for those of you that are too young to remember that they existed. But anyway, so the point here is that the red bordered circles are the kipukas. So if we look at the allozymes in particular here, what you see is along the same distance, if they're separated by lava, you get really marked separation, genetic separation between these little islands of vegetation. And this is really remarkable because these pockets are only, 100 or so years old. So what is actually going on here? And these are the spiders that are inside versus on the edge. So I just want to note just briefly here, this is a study just recently conducted by a postdoc in my lab and who came back and worked on this. And he was interested in just, to what extent is this a universal phenomenon such that these kapukas just generally isolate things. And so he did a metabarcoding study with others in our lab. And if we focus first of all, on the center of the kapuka, this is what's really isolated. And what you see in the center of the kapuka, you get this very marked area effect. So the bigger area, the more OTU richness. Here, we're simply looking at genetic entities. We're not looking at what the species are, we're simply looking at genetic entities. And the ZOTU, which is the top line, is the more finely divided zero radius OTU. It's just the actual genetic entity, whereas the lower bar is clustered. So you see, you get a very marked area effect that's even stronger when you look at just the zero radius OTUs. So a very marked area effect that you do not get when you look at the edges, which the edges are basically, it's not an island for the edges. So this is even more fascinating in that he separated out the different orders here. And what you can see, if we just look at the purple lines, what you see, they're just flat, and you say, well, there's no relationship there. What this is showing, actually, this is between the turnover between these little kapukas, these little islands of vegetation. And what it's showing is, at least at the ZOTU level, and very high when you cluster the OTUs, basically you get almost complete turnover as you go from one kapuka to the next. When they're clustered, you see that you still get marked differences between these kapukas, but it's accentuated when you look at the ZOTUs. And so you're thinking, whoa, what's going on here? And what this clearly shows is that the kapuka are creating different combinations of taxa. They're different sizes, and so they're different isolations. So you're going to get different groups of taxa in any kapuka, but also you're getting genetic differences between populations, which is making the ZOTU values higher than the clustered OTUs. So this is just an example of what you can do, in these very young habitats. I want to just finish off with what we can do with islands that are slightly older. And so we're going the very youngest stages of on these hotspot archipelagos, you're just getting population divergence. It's just starting out in the within an adaptive radiation. So what happens as you go one step older? So one step older, and you can actually look at the whole diversification process when you do this. So I want to just quickly show you one example, and this is from spiny leg tetragnatha. These are long-jawed spiders that we've studied across the island chain, and they show repeated evolution of ecomorphs. So on any, this is the oldest island where you get a big brown one, a green one, and a little brown one, and you get repeated evolution of these forms, including a maroon one as you go down the archipelago. So we're going to focus on this little patch of the youngest island, the youngest part of Maui, where you get all of these species living together. And so you say, well, what's going on? So this is the work then of and I'll just, these are the ecomorphs, just so that you don't think that I'm just kind of playing around with you. The green ones on different islands look almost identical. These ones are not related, rather they're related within an island. This is a maroon one on Oahu, a maroon one on Maui, and a big brown one on Kauai, a big brown one on Maui. This is a little brown and a little brown. But you see the ones on the right, you know, these ones in particular, these ones here, they're each other's closest relatives, whereas this one looks almost identical to this one, but it's not related. So you get this repeated evolution of ecomorphs. And so what we say, okay, what's happened on the youngest island? On the youngest island, there's just a population from Maui. So you say, well, you know, that's of limited interest. What's happening on Maui? And so what's really cool here is that actually you get 3 species of the green ecomorph on Maui, on the East Maui. And so these are parapatric. This is work I did quite a while ago. They just kind of are arranged across the island, the flank of the volcano. And so Darko Kataris, who is a student in my lab, he wanted to see what in the world is going on with these things. You know, are they really different? What's happening? So Darko used this axon capture approach, which I won't go into detail in at all. But the point here is that he showed that each species was very distinct, and each population on the different volcanoes was very distinct. So what it seems is that you've got a broad, you know, a broadly distributed taxon, you know, across here you see Hawaii, Maui, Lanai, not on Molokai for whatever reason, but anyway, you've got them on these islands. It seems that you get divergence such that there was a new species on Lanai, a new species on the Big Island, and then they seem to come back so that you get all three species occurring together on this one volcano of East Maui. So what in the world is going on? So Darko looked at this further, and what he showed was that actually, you know, they're very distinct, but you also get little pockets of distinctness, even, you know, within that range. And so what seems to be happening in a nutshell is that you get isolation. You know, each of these species is related to something that is isolated somewhere else. It seems that you get isolation, you get secondary sympatry resulting in interaction, and then subsequently ecological divergence allows stable co-occurrence. So is that you know, is that what's happening? And if it is, what's the isolating factor? So this is, I have a student at the moment who's working on what is the isolating influence here, and just super quickly, what she's been looking at methyl ethers on the webs of the spiders. These spiders are wet and they're in rainforest, and they have minimal courtship. They just run up to each other. You can put them in a vial together and shake them and they mate. This is the male and female mating there. So it's just a very kind of, you know, no messing around, just kind of they mate. And so Ashley was interested in the methyl ethers and how distinct they were. And so she looked at this specific one area on Maui, and high elevation and low elevation. And what she showed was this is high elevation, and you see each species indicated by the pink arrows, each species is totally unique in terms of the methyl ether composition on their silk. Then you look at lower Waikimoi, you find that each species is completely distinct. But you see, there's quite a lot of overlap. When you put all this data together, there is overlap, but these are among species that don't co-occur. And so there's obviously a lot more to find out here, but you can start to see what the isolating factors might be. So with that, I just want to summarize where we're at. I talked initially about how species multiply through allopatric, parapatric, potentially sympatric speciation. But the big thing I wanted to emphasize was the use of the island chronosequence that provide just perfect systems for looking at different stages of the adaptive radiation process. They highlight, so far the data has highlighted the role of isolation in initiating divergence. But it can also show you that it can provide insights into what happens when taxa that have diverged in isolation come into secondary contact. The biggest thing, most likely when they come back together, they're going to go extinct. And I've got a student currently who's trying to understand the extent to which extinction might be involved in this initial secondary contact time. But under some situations, it seems that you can get character displacement, because in each island, you get these different ecomorphs that are each other's closest relatives. And from what Ashley showed, there's also reinforcement in terms of the methyl ethers and the distinct chemistry that each species has when they co-occur at a site. And with that, I'll just say thanks to you and many students, postdocs, and collaborators in the lab. And yeah, thanks for listening. And I'd be delighted to, with Clarisse, we can have some questions, and I'll stop sharing here.
Great, thanks very much. This was super interesting. If anyone has a question, please just put it in the chat. There is already a question there. I don't know, Rosie, if it helps you to read it yourself,
let's see,
but I'm going to read it.
Perfect, yes.
Would you expect that the meta-population and community dynamics produced by lava flows to be equivalent in new and old islands? So he's thinking of rejuvenation volcanic activity for the older islands. In other words, what happened on the Kipuka around Diamond Head in Oahu or Kilauea crater in Kauai?
Well, these are excellent questions. Darko was the one I highlighted who'd done the initial work looking at the exon capture data for these divergent taxa on Maui. So yes, what we're trying to figure out now is to, given that you've got the initial fragmentation if Hamp Carson was right about the crucible of evolution, where you get these different the lava flows resulting in the initial divergence. If that is the crucible, you'd expect to find signatures of those of divergence as you go from older islands. I mean, it's going to slowly erode, but what we need to do now is look at older islands and see whether, if you look at lavas of different age, whether you get disjunctions. And I should say, I was just talking to Carrie Shaw about this, and it seems that maybe you get that in cricket. So anyway, it's going to be quite exciting to see that.
Very nice. Then Jimena also has a question. What evidence is there that the methyl ethers in the webs mediate mate choice and or pre-mating isolation?
That is a perfect question. And I wish I had the video. So Ashley, who I mentioned, who's done this work, she started this because she had exactly the same question. What is the mechanism of recognition? So what she did was use the silk of the two different species. And so basically she asked the male, which one do you prefer? And I wish I should, if you want the video, I'd be happy to share Ashley's video with you, but it's beautiful because the males go up to the silk, test it, just, which one is the right one with their legs, because that's where their sensory apparatus, for smelling apparatus is. They test it, and then they go up the one that's of the same species. She also, in case it was actually the silk itself that was leading to recognition, she extracted the methyl ethers and put them on threads and did the same choice experiment. And again, they tapped each thread and went up the right one. Just beautiful evidence of that they're actually selecting these methyl ethers.
Jimena, and I agree. This is so cool. Thank you, she writes. Okay, I think this was an excellent last question about your talk. So now I suggest we switch to the general discussion. Clarisse and Rosemary will answer some questions generally about the role of geography in speciation. So if you have questions about that, please continue posting them in the chat. And in the meantime, I will kickstart this session with a question about how important